In this video, we'll be talking about how information travels through the body through neurons, and within those neurons, it's through something called action potentials. And before we can understand action potentials, we have to understand what passive transport and active transport are. So, passive transport is moving from high concentration to low concentration. And that is a naturally occurring process. It does not require energy. It's actually a major point. It does not require energy, and it's going to occur naturally. Doesn't have to be forced to happen. Think of when if you're in a room with somebody and somebody starts smoking, and you're on the other side of the room, you're eventually going to smell it because the particles in the smoke in the air spreads out. It doesn't just hover around the person that's smoking. It spreads out. That's why there's no longer smoking sections and non-smoking sections inside of restaurants, because it didn't make a whole lot of sense for when the particles are going to spread out and the smoke's going to spread out. So passive transport is when something spreads out. It moves from high concentration to low concentration. Active transport, on the other hand, is when we're moving from low concentration to high concentration. This is not a naturally occurring process, and since it's not naturally occurring, it has to be forced to happen. And we can force it to happen through inputting energy. And recall that the cell energy is going to be ATP, adenosine triphosphate. And different types of passive transport. There's going to be different types of passive transport involved in the sodium potassium pump and action potentials. And so there's simple diffusion, where something just spreads out and there's no barrier between things. So simple diffusion, things just spread out and it's not traveling through anything particularly, it's just spreading out where it's at. Facilitated diffusion, on the other hand, is it is still passive transport. It does not require energy. It's a naturally occurring process, but the particles that are spreading in or spreading out, they are doing so through a protein channel. And basically, some particles cannot freely pass through a cell membrane. This graphic right here shows you which particles can and which particles can't. But keep in mind that some particles can't. So like here are some examples of particles that cannot pass freely through a cell membrane. And when a particle cannot pass freely through a cell membrane, even if there's a low concentration over here and a high concentration over here, they're not going to be able to pass through. Unless we have some sort of protein channel here where the particles, it acts like a tunnel, and the particles can then just flow through it in order to spread out. So that's what's happening with facilitated diffusion. Is there's a transmembrane protein that works to spread things out, or that allows certain particles to flow through it in order for them to spread out. And then osmosis, that's just the passive transport of water specifically. Active transport sometimes involves these transmembrane proteins as well, except we call them carrier proteins for these, because instead of just providing a tunnel for things to travel through, it's actually it's kind of like a tunnel, but it has like spots where things can bind to it, and then once the correct number of particles are bound to it, it's going to carry the particles across. So instead of just naturally letting things flow across, it's going to carry the particles across and change conformations. And when it's doing so, it has to do it this way because it's flowing things from low concentration to high concentration. And when things are flowing from low concentration to high concentration, that is not a naturally occurring process. And since it's not naturally occurring, it has to be forced, and we can force it to happen through these carrier proteins, where they're going to carry things, up the concentration gradient, so from low concentration to high concentration, and doing so requires energy in the form of ATP. So both of these things are going to be involved in both of these things are going to be involved in the sodium potassium pump and action potentials. So we have to absolutely understand these concepts before we can even begin to understand the sodium potassium pump or action potentials. So when we talked about how things like to spread out, that's just a naturally occurring process. It's called diffusion. Keep in mind that like particles like to spread out. So if I have a bunch of sodium particles here on the outside of the cell, which is true, sodium is high on the outside of the cell. If I have a whole bunch of sodium particles, they're going to want to spread out because they are all like particles, but also because they all have a positive charge. So something that's important here in the sodium potassium pump is that there is an electrochemical gradient. So not only do we have like particles, like chemicals that want to spread out, but also like charges that want to spread out. So we have two different things, all both acting to cause these particles to spread out. So an electrochemical gradient is the difference in both charge and concentration across a membrane. And within the sodium potassium pump specifically, it is an antiporter, which means there are two particles moving in opposite directions across a carrier protein. So I think one of these, no, maybe none of these are indicating the sodium potassium pump, but the sodium potassium pump, we're going to have 3 sodiums and 2 potassiums. So 3 sodiums are going to move outside of the cell for every 2 potassiums that are going to move inside the cell. And they kind of do so simultaneously, both going against the concentration gradients. And you guessed that if it's against the concentration gradients, then it's going to require energy. All right. Talking about the sodium potassium pump. Again, the sodium potassium pump is an antiporter, and it is moving sodium out of the cell and potassium into the cell. Both of these movements of sodium out of the cell and potassium in the cell are against the concentration gradients of these two chemicals. So it requires energy. Let's take a look at this graphic right here real quick, where we see that potassium is naturally high on the inside of the cell. We're not going to be talking about these organic anions too much, but they are contributing to the cell membrane voltage, or the cell voltage as well. But we're primarily going to be focusing on potassium, chlorine, sodium, and calcium here. So we said that potassium is high on the inside of the cell, so it wants to flow out. But this sodium potassium pump, it is pumping potassium into the cell. In fact, it's pumping 2 potassiums into the cell for every 3 sodiums that it's pumping out of the cell. So we have this ratio of 3 to 2. That's going to be good to remember. And so potassium's high into the cell, sodium's high outside of the cell, and we're pumping sodium out of the cell to create even more of a difference in membrane potential. So we already have a difference in sodium and potassium across the membrane, but we are moving these things against their concentration gradients to make even more of a difference. So we have this electrochemical gradient, and we're making it even more so of a difference in membrane potential. So what is the purpose of the sodium potassium pump? The purpose of this is to create an electrochemical gradient so that sodium will flow into the cell when it is able, and potassium will flow out of the cell when it is able. So we're creating this gradient, and then we're doing so that when it gets to a certain point and we want to allow sodium to flood through, so we want to open up a channel that allows sodium to flood through, and so that it can flow into the cell where it wants to be, it's going to do so very rapidly and change the voltage of the cell very rapidly. And we're going to be talking about how that is going to contribute to an action potential forming. And then also involved potassium, a potassium channel will open as well to bring it back down to its resting potential, but we'll get there. But let's keep in mind these differences in concentrations inside and outside the cell. So maybe pause the video and try to remember these. Potassium is high inside the cell, and then all the other ones we're dealing with are high outside the cell concentration. So potassium, high concentration inside the cell. Sodium, chloride, and calcium are high concentrations outside the cell. Potassium's high inside the cell. Sodium, chloride, calcium, high outside the cell. So now we're going to be looking at what an action potential is. Now we know that the purpose of the sodium-potassium pump is creating this difference in concentration of both these chemicals and their charges across the cell membrane. And the difference in charges across the cell membrane creates a difference in a voltage. And then we have this graph that we can kind of measure that at different points in time. So the cell has a resting membrane potential of about -70 millivolts. And this is closer to potassium's resting membrane potential than sodium's because there's more potassium leak channels than sodium's. But we're not going to really talk about leak channels here because they play a role, but they're not like the main players. So we're primarily going to be talking about the different channels we're going to be talking about are voltage-gated ion channels and ligand-gated ion channels. We're going to see what their role is here. So we're going to talk about, so we have the resting membrane potential is about -70 millivolts, and that we don't really need to know why. That's just what it is. And basically we have this action potential, and that's how signals travel through the body. They travel through neurons. Information in the body travels through neurons. We're reading this bullet point right here. Information in the body travels through neurons, and within these neurons, information travels as action potentials. Action potentials are all or nothing, which is very important to remember. So we can't send a partial signal. So action potentials are all or nothing, meaning there will either be a full action potential or no action potential at all. You cannot send a partial signal. And this goes for anything in the body. Action potentials are going to cause my muscles to contract if I try to lift something up. And I can't only partially cause my muscles to contract. It's an all or nothing thing. Or if I feel pain, I can't feel less pain by sending less of an action potential. Either I'm sending full action potential or not a full action potential. And the way our body gets around that is we're always going to be sending a full action potential, but maybe we just don't send the action potential across as many neurons if we don't want as strong of a signal. So that's what an action potential is. It's just a way that information travels through neurons in the body, and we're going from neuron, and we'll show down here below how we can go from neuron to neuron. But now the purpose of the different ion channels, we have the purpose of ligand-gated ion channels is to get the voltage to the threshold. So we have this resting membrane potential of approximately -70, and we need to get it to -55. That is the goal. If we can get it to -55 millivolts, that means we are going to end up having an action potential, and we'll show why here in a little bit. But if we can get it just to this amount right here, we're going to end up getting it all the way to this amount and create the action potential. But if we don't quite get that far, let's say we only get to here, it's just going to go back down. It goes back to the resting membrane potential. If we don't get to threshold, then there's not going to be any signal sent. That's very important to remember. If we don't get to threshold, there's no signal sent. So the ligand-gated sodium channels, they are going to be helpful towards getting this to threshold Once it gets to threshold, it's going to go up, and then we'll talk about what happens after that. But once it gets to threshold, we will get an action potential. But if it does not quite get to threshold, if we don't have enough sodium enter the cell through these ligand-gated sodium channels, then it could get really close, but if it doesn't get to threshold, it's going to go back down to the resting membrane potential. We also have some of these ligand-gated channels. They actually have a purpose of inhibiting a response. So sodium's purpose is an excitatory. It's triggered by excitatory neurotransmitters, and they are going to increase the likelihood that we get to threshold. But we have other neurotransmitters that allow potassium to leave the cell or chlorine to enter the cell. And both of these are going to decrease the likelihood of getting to threshold. So when potassium leaves the cell or chlorine enters the cell, the charge of the cell is going to decrease. And look, we are even further from a threshold. So now a threshold is really unlikely to happen. And I'll show what that looks like in our pictures down below. So also I wanted to point out that this action potential graph, this is really taking place at the axon hillock. And the axon hillock is a really important region of a neuron. But we'll talk about that down here. All right. We're going to first go through an action potential for a cell that has an excitatory response. So we just had an action potential travel down an axon. So we are at the presynaptic neuron here. Presynaptic neuron, presynaptic cell. These are just types of cells. Presynaptic neuron. And then we have the postsynaptic neuron. And the reason why we call it a presynaptic neuron and a postsynaptic neuron is because this region right here is called the synapse. The synapse is the region between two neurons, and that's where neurotransmitters are flowing around. And we'll show that here in a little bit. But on the presynaptic neuron, let's say that it already had an action potential, and that action potential is traveling down the axons. I don't have the myelin sheath drawn in yet or here, but just imagine they're there, or maybe I can just leave that there. But we have this action potential that's traveled down, and it has now reached the axon terminal. The axon terminal is just the end of the axon. And once the action potential reaches the axon terminal, it triggers calcium channels to open. So I'm going to do those in pink here. And we have these calcium channels right here. And when they notice that there's an action potential that has reached the end of the axon, they are going to open. And then calcium is going to flow from high concentration to low concentration. If we recall, calcium is high on the outside of the cell. So if these channels open, think about which direction the calcium is going to flow. If there's a lot of calciums out here and not a lot of calciums in here, that means calcium is going to flow in this direction into the cell. So calcium is high on the outside of the cell, and it's going to flow into the cell because it noticed that the action potential has reached the axon terminal. Calcium plays a really important role here because when calcium enters the cell, we have these neurotransmitters that are contained inside of these, let's just call them globules. And these neurotransmitters, they are just hanging out here until calcium enters the cell. When there's a high concentration of calcium, calcium is going to interact with some things that cause these neurotransmitters to be pulled in this direction. If you want to know the details of that, that can be covered later, but it's not important for this class. But when calcium is present, it causes these neurotransmitters to move in this direction. So moving to the direction of the end of the axon so that they can be released. So let's see what this looks like with one of these. Let's zoom in and figure out what's going on. So let's also keep in mind, I didn't draw that here and I totally should have, but the cell membrane is made up of the same materials that the outside of this neurotransmitter is made out of. So when this neurotransmitter fuses, or it reaches and touches the end of the cell here, touches the cell membrane, it's going to fuse with it and allow the neurotransmitters to be released. So let's look at what's going on. The neurotransmitter, we just said it is moving in this direction. So let's draw what it would look like once it reaches the end. So we now have the neurotransmitter globule is binding to the end of to the edge of the cell, and we have the neurotransmitters inside, and then also we now have it fused with the membrane, and when it fused with the membrane, it's allowing the neurotransmitters can now be released. So these neurotransmitters are now released into the synapse, which again is the region between the cells. And now they're just bouncing around, flowing around in the synapse. Something that this postsynaptic neuron has that's very important is it has receptors here. And these receptors, these are neurotransmitter receptors. And when the neurotransmitters, they're just bouncing around, and eventually one of them is going to land on a receptor. These receptors are connected to ligand-gated ion channels, and initially they're closed. But when a neurotransmitter binds, it's going to cause the receptors to open. And when the receptors open, these ligand-gated when the receptors bind a neurotransmitter, these ligand-gated ion channels are going to open. And when the ligand-gated ion channels open, it allows whatever they're specific for is going to flow either in or out of the cell. If these neurotransmitters are excitatory neurotransmitters, that means they are going to allow sodium or some other positively charged ion. It's typically going to be sodium, but they're going to allow sodium to enter the cell. And the important thing is that it's a positively charged ion, because when a positively charged ion moves into the cell, that means the cell is going to have more positive charges. And let's look back at our graph up here of an action potential. In order to get to threshold, we need more positive charges inside the cell. We need the inside of the cell to become more positive so that we can get to threshold. And once we get to threshold, there's going to be an action potential, and we'll talk about why that is. But so sodium flowing into the cell is going to help us reach an action potential. And let's give a little more details as to why. Let me zoom in a little bit. So we have the presynaptic neuron, an action potential traveled all the way through. Once the action potential reaches the end, it triggers calcium to flow into the cell. Once calcium flows into the cell, it signals these neurotransmitter globules to move to the end of the cell fuse with the membrane, and then release their neurotransmitters into the synapse. Once the neurotransmitters are released into the synapse, they are eventually going to be binding to these the receptors, the neurotransmitter receptors. When they bind to a neurotransmitter receptor, it causes the ligand-gated ion channels to open, and whatever cell or whatever ion, it's ligand-gated ion channel because it's a channel that allows ions to pass through. And when it opens, it allows whatever ion is associated with that neurotransmitter, or more specifically, associated with the ligand-gated ion channels, it allows that ion to move through the cell from high concentration to low concentration. So if that ion is high in concentration on the outside, that means it's going to flow into the cell. If that ion is high concentration on the inside, it is going to flow out of the cell. In this case, we are saying that this one is going to trigger sodium to be released, a sodium channel to open. And we already know that sodium is high on the outside of the cell. So sodium, when it flows in, when this channel opens, sodium is going to flow into the cell. And I'm going to represent sodium as these red circles with pluses. And look what's happening. We have a bunch of sodium. It's flowing in through there, flowing in through here. So we're getting a whole lot of sodium inside the cell now, because these channels opened, and now the sodium is flowing from high concentration to low concentration, and it's flowing into the cell as fast as it can. And think about this. Is the sodium just going, we're getting a very high concentration of sodium in this region of the cell now. Is the sodium just going to stay there, or is it going to want to spread out? Well, it's going to want to spread out through diffusion. And as it spreads out, it's going to be moving across the cell, and it's going to keep spreading out until we get over here at this region right here that I highlighted. We call this the axon hillock. All right, the axon hillock is it's important because it is a region of the cell that has a very, very high concentration of voltage-gated ion channels. So both voltage-gated sodium channels and voltage-gated potassium channels. Those are the two key players here. And let's see what happens. So we have sodium has now entered the cell because there was a neurotransmitter that binded to a receptor that controlled a ligand-gated ion channel. If I didn't mention this before, a ligand is just something that binds to something else. So it's ligand-gated because it's closed until a ligand is bound to the receptor. And when a ligand is bound to the receptor, it opens the gate. Once the gate is opened, sodium flows, at least in this example, sodium flows in, and it starts spreading out. And so right now, or at first, we have a very high concentration of sodium here. So the charge of the cell is very high and is very more positive here, it became more positive. And then it's going to spread out to where now we're going to have a more positive charge that's throughout the cell, not just in this region. And let's look at this region right here specifically now. The sodium has now spread out to where it is now it has now increased the charge of the inside of the cell here at the axon hillock, which that is where this graph that is what this graph is showing. This is the action potential at the axon hillock. And we just said we are now at the axon hillock, and we have more positive charges that have now entered the cell. So more positive charges going to cause the voltage to increase, And if we have enough sodiums, we might get to threshold. If we get to threshold, then there are other transmembrane proteins. There are, we just talked about the ligand-gated ion channels. But now, once we get to threshold at the axon hillock, we're going to open other types of sodium channels called voltage-gated sodium channels, or sodium voltage-gated ion channels. And so once we get to threshold, if we have enough sodiums flood in here, if enough sodiums flood into the cell to where they can spread out and get a big enough charge in this region here, then we are going to get to threshold, and that's going to cause these other ion channels located here in the axon hillock to open. Let's zoom in and take a look at what those look like. So the axon hillock is important because we have a very high concentration of voltage-gated sodium channels and voltage-gated potassium channels. So these voltage-gated sodium channels, let's draw them in orange here. They're going to open when we get to threshold. And there's a whole lot of them. All right, they open when we get to threshold. Let's say we got to threshold. There were enough positive charges here that traveled across the cell, and the inside of the cell is now it became positive enough or less negative enough to reach threshold. And once we reach threshold, it's going to open these voltage-gated sodium channels. Let's talk about why they're voltage-gated sodium channels. So we said ligand-gated sodium channels open. They're like gates that are closed, but they open when a ligand binds to the receptor. Voltage-gated sodium channels, they are like gates also, except they open when the voltage gets to a certain amount. And that certain amount for this voltage-gated sodium channels is negative 55 millivolts. So when we get to negative 55 millivolts, these sodium channels, these voltage-gated sodium channels are going to open, and sodium is going to rush into the cell. When sodium rushes into the cell through these, there's a whole lot of them. There's trillions of these in the axon hillock. And there are so many of them, it's almost as if we don't even have a cell membrane here. The sodium is just flooding through very, very, very fast. So let's look at our graph again. Notice how the diffusion of sodium caused us to slowly go up. But once we get to threshold, it's going to boom up really fast. And because sodium is flooding into the cell very, very, very fast. So this action potential, it has now been reached, and the action potential is going to travel across the cell. Once we get to a charge of approximately positive 40 millivolts, we have now reached the action potential. It's high enough of a charge to where now we can close those sodium channels and start pumping the sodium out of the cell again through the sodium-potassium pumps and get back down to a resting membrane potential of about negative 70 millivolts. But to help for that to happen, when we get to the top here, let's look. I'll draw this in orange. When we get to the top right here, the sodium channels are going to close. Specifically, the voltage-gated sodium channels are going to close. So the voltage-gated sodium channels are going to close, and it says potassium ions out. That's because the potassium channel, voltage-gated potassium channels are going to open. So the voltage-gated sodium channels, they open when we get to negative 55 millivolts. The voltage-gated potassium channels, they're going to open when we get to about positive 40 millivolts. And the purpose of that is to bring the charge back down. or the voltage back down. So the voltage-gated potassium channels are now going to open, and let's just draw these in purple. And so we said that the voltage-gated sodium channels is going to cause sodium to flood into the cell. So we've got a lot of sodium flooding into the cell. And the voltage-gated potassium channels now, they're going to allow potassium to flood out of the cell. So again, we can't just have sodium flood out of the cell to bring it back down to normal. We can pump sodium out of the cell, but remember, sodium is high on the concentration on the outside of the cell. So it's not just going to naturally flow out of the cell. If we want to bring the charge of the cell back down, the easiest way to do that, and the most energy efficient way to do that, is going to be to pump potassium into the cell. So that's what's going to happen here. At the top here, we're going to have potassium is going to, voltage-gated potassium channels are going to open. All right, voltage-gated potassium channels open. And when the voltage-gated potassium channels open, again, potassium I misspoke earlier. Potassium, so sodium is flowing into the cell when the voltage-gated sodium channels open. When the voltage-gated potassium channels open, we're going to bring the voltage back down to negative 70, ideally. And we're going to bring the voltage down by making the inside of the cell more negative. And we can do that by having these potassium ions, they can flow out of the cell. So the cell can become more negative by more negative charges entering or more positive charges leaving. In this case, we've got a lot of potassium ions in the cell, and potassium is going to want to flow out of the cell. And it's going to do so through these voltage-gated potassium channels that just opened because we reached the desired voltage for them to open of positive 40. When they open, potassium's going to rush out of the cell and make the cell more negative. And so it's going to make the cell more negative, and actually it does too much almost. So it gets past negative 70. So it's actually it opens and then it gets to negative 70, and then it keeps going. It actually does a little bit too much, and it gets down to maybe negative 100 or something. And we call this hyperpolarization because polar means that there's a difference in charge across the membrane. Depolarizing means we're decreasing the difference in charge across the membrane. Depolarization. And then hyperpolarization is we're increasing the difference in charge across the membrane. So think of negative 70 as the resting membrane potential. And so this is, since there's a difference in charge, it's a difference in charge of negative 70 millivolts. Since there's a difference of charge, it is polar. And if we decrease that difference in charge, it's depolarization. When we increase that difference in charge, it's hyperpolarization. And so potassium leaves the cell, and so that to bring the resting membrane potential back down to negative 70 so that we can have another action potential soon after if we need to, or there will be typically. And then it kind of messes up our concentrations of sodium and potassium. We now have a lot of sodiums that enter the cell, and they end up getting pumped out through the sodium-potassium pump, which was explained up here. So now we're going to start requiring energy to pump sodium out of the cell and potassium into the cell to get rid of all that sodium that we just let in and potassium that we just let out. So get rid of all the sodium that we just let in and then bring back all the potassium we just let out. So that's where the sodium potassium pump is going to come in, and the action potential, we had opened the potassium channels, and when we did that, we left them open a little bit too long, and it caused hyperpolarization to occur. We went a little bit too far, and but that's okay. We're just going to now close the potassium channels. So let's actually label that. Voltage gated potassium channels are going to close here. And when they close, the cell is now going to start, we have different leak channels and different things, and the cell is eventually going to get back to this negative 70 value, which is its resting state, from both the sodium and potassium channels being closed. Once we get back to the resting membrane potential, we can have another action potential, and these action potentials are just going to keep occurring through this entire axon until they get to the end. And when we get to the end, they're going to release calcium, and the calcium is going to cause neurotransmitters to be released, and then those neurotransmitters can either cause an excitatory or inhibitory response on the next action potential. We're going to be talking about the other examples of this here in the next video, where we'll see what happens if these neurotransmitters bind to a receptor that causes potassium to leave, or and then the final example is going to be an example where we have neurotransmitters that binds to a receptor that causes chlorine to enter. So we'll explore what happens in these two inhibitory responses in the next video. But I think that's everything I wanted to cover. I might not have pointed out what the purpose of these insulators called myelin sheath here, what their purpose is, but their purpose is to speed up the action potential. So the action potential is traveling through, and the myelin sheath here, they insulate the axon so that you're not losing any of the action potential dissipating out. And we also have these nodes of Ranvier here. I'll just label these both. So these are myelin sheath. And their purpose is to speed up the action potential. And then here, these gaps between the myelin sheath are called nodes of Ranvier. And what they're kind of like the axon hillock in that they have a high concentration of voltage-gated sodium and potassium channels to kind of re-strengthen the signal whenever we get to them. So re-strengthen the signal here, re-strengthen the signal here, so that we don't, so that the signal is not lost by the time we get to the end, because we need a full signal to get at the end so that calcium rushes into the cell. But I think that's everything that I wanted to cover. We'll cover this is an excitatory response here. This first example is an excitatory response. We're going to go over two inhibitory responses in the next video or two.